Hey team, okay, this week we are all about cells, right? So look how far we've come. We've talked about what makes up molecules, the types of bonds that make up molecules. We've talked about how those different bonds give molecules different characteristics. And then we talked about the specific molecules in cells, right? All those biological molecules. We talked about proteins, amino acids. Yeah, proteins that are built up out of amino acids, nucleotides that make up nucleic acids, right? Carbohydrates, fats, lipids. We've come a long way. Now we're going to put all of that together because that all helps us understand how cells function. Because cells are made up of all of those macromolecules that we talked about that have very specific chemical properties based on the bonds that are in them, right? Kind of see how we're kind of layering that up? So we're going to talk first about prokaryotic cells, and then later we'll deal with what's called eukaryotic cells, okay? So let's jump in. Okay, so let's way on back into that first week. That feels like it was almost 20 years ago. But that first week of our class, we talked about the idea of the cell theory, right? So all living things are made up of cells. The smallest unit of life is a cell, so if it's something smaller than a cell, we do not consider it alive. It does not have those characteristics of being alive if it's smaller than a cell. And new cells come from old cells, okay? So life does not just spontaneously pop into existence. New cells are basically the offspring of previously existing cells. Okay, so it's kind of our basis for this week. Is all living things are made out of cells. Now let's talk about what types of cells are there, how are those cells different from each other, and do different forms of life have different types of cells? Okay, so all cells share four basic things in common. Okay, all cells will have some sort of plasma membrane, some sort of outer covering around them, right? That's made up of the molecule that we talked about, the phospholipid molecule, right? Plasma membrane's a phospholipid bilayer, some extra stuff on there, some extra icing if you would. All cells have a cytoplasm. So the cytoplasm is everything inside of the cell. And this is where we have all of the organelles that are suspended, right? All the cellular machinery, all the DNA, everything that's going on, all those enzymes that are working, those are hanging out in the cytoplasm. So it's a thick, viscous fluid. It's roughly 70% water of the cytoplasm. The fluid that fills up the cytoplasm, we call it the cytosol, okay? It's basically just like the jelly that fills up a cell, okay? It's a mixture of water, carbohydrates, proteins that are all kind of in there. It's where everything that's going on inside the cell is happening, basically. So all cells have some sort of cytoplasm. All cells have ribosomes. Ribosomes are an organelle that are used to construct proteins, okay? We talked about ribosomes briefly when we talked about the types of RNA. So ribosomes are made out of RNA, and they build that peptide bond between amino acids. All cells have DNA. All cells need some sort of genetic material to, one, give them instructions for how to build all of the molecules and proteins within their cells, but two, that DNA is also what's sent down to the next generation. Every time that cell divides, it's going to send on its DNA to the next generation. Okay, so all cells have DNA as well. So in our big, beautiful, complex world, we've only found two basic categories of cells. All life on our planet is either going to be a eukaryotic organism or a prokaryotic organism, right? So us, and plants, animal fungi, and some other weirdos called protists are eukaryotes, okay? Things like bacteria and other organisms similar to bacteria called archaea are prokaryotic cells, okay? So what are some of the differences that we notice here? Well, first, size. These aren't really drawn to scale, but prokaryotes are much, much, much smaller than eukaryotic cells. And look at what's inside of them. There seems to be a lot more going on inside of that eukaryotic cell, right? Eukaryotic cells have many organelles, which organelles are mini organs, which is what we'll talk about a little bit later, but they're specialized structures within that cell to carry out specific functions. Just like we have specialized organelles inside of our bodies to carry out specific functions, cells have specialized structures inside of them to carry out certain functions for them. Okay. We don't find those organelles in the prokaryotic cells though. Okay, so we do not find specialized organelles inside of our prokaryotic cell. Prokaryotes are basically just a membrane, cytoplasm, some DNA in a nucleoid region, not encased in a separate membrane. So their DNA is just floating in the cytoplasm, not separated by anything, and ribosomes. So prokaryotic cells are much smaller, and they don't have as much cellular machinery or specialized compartments within their cell. So again, in our 3 domains of life, so 3 major branches, groups of life, these are the biggest categorization that we can have. These are the most inclusive domain in our taxonomy level. Eukarya is so named because these are organisms that have eukaryotic cells, those type of cells that have many organelles in them. Archaea and bacteria have prokaryotic cells, so they're much smaller. They're basically just a cell membrane, cytoplasm, DNA, and ribosomes. So prokaryotes are absolutely tiny. There's no way we can see them with the naked eye. Some single eukaryotic cells we can see with the naked eye, even though they're still really, really tiny. But single prokaryotes, definitely we could not see a single prokaryotic cell with the naked eye. So most bacteria, most prokaryotic cells are in the range of 1 to 10 micrometers. To give a sense of scale, the average human cell is right around like 60 micrometers. Okay, so these are some cheek cells that we've taken from inside of our mouths and we've stained them blue, right? Your cheeks aren't blue. This is a stain that we added to them. And what we find is, here's our cell membrane of the cheek cell. This is our cheek cell right here, human cheek cell. Here's the nucleus where the DNA is at, and this is all the cytoplasm in here, okay? These tiny little dots on that cheek cell, those are bacteria, okay? So those are prokaryotic cells. So it gives you a sense of scale of how tiny they are, okay? They're about 50 to 60 times smaller than the average eukaryotic cell. Really, really, really tiny, okay? And I just love this image because it gives you another sense. Here's a tip of a pin, okay? It's been colorized, scanning electron micrograph colorization, and in the tip of this pin, we look at, just in that one little section right there, look at all of those bacteria, okay? So they're really, really, really tiny. There's probably more species of prokaryotes, though, than the rest of the eukaryotic tree combined. So they're incredibly diverse. They're everywhere, all the time, even though they're so simple. But they're one of the oldest forms of life on our planet. Okay, what do they look like? So prokaryotes come in 3 basic cell shapes. Okay, so all of the millions of species of prokaryotic organisms come in basically 3 shapes. So they can come in a spherical shape that we call cocci, a rod shape that we call bacillus, or a spiral shape that we call spirillus. Okay, so that's not a super helpful trait when we're trying to identify a specific prokaryotic organism, because there's only 3 shapes and there's millions of prokaryotes, right? But for some reason, we find that they only come in one of these 3 shapes. So when we're looking at prokaryotic cells, and if we want to try and identify what bacteria or prokaryotic organism are we dealing with, we'll often look at things like cell shape, as well as colony shape, colony color, colony size, the borders of the colony. Can it metabolize certain sugars? Can it metabolize certain, and things like that, right? So it's usually easiest to do genetic sequencing on prokaryotes to figure out which species we're looking at, but it's good to know that they only come in one of these 3 shapes. Okay, so let's dive into a prokaryotic cell and talk about what's going on in here. Okay, jumping into the inside of a prokaryotic cell. Here we go. So we're going to talk in very general terms. Again, prokaryotes are very diverse. There's millions of species of these guys, but they all kind of share the same basic common characteristics. They all have 1 chromosome that they keep in kind of a region that we call the nucleoid region. It's just the area where the chromosome kind of condenses, but it's not enclosed in a secondary membrane. Okay, so we'll see what that looks like when we talk about eukaryotic cells, but it's just free floating in the cytoplasm, basically. Also, the bacterial chromosome is one big circle. So it's one big circle of DNA, and they only have one chromosome. Okay, so it looks like it's like all kind of a string of yarn right here, all wound up. That's because it is. It doesn't exist in that one big circle in the cytoplasm, but if we were to expand it out, untangle it, it would be one big circle. So that's hanging out there. So that DNA contains the instructions for how to make all of the proteins that it might need, any enzymes that it might need to digest or change around molecules, how to maybe be resilient to things like antibiotics could be contained on that chromosome. The other major component of bacteria is their ribosomes. So all these kind of dark brownish dots right there, those are all ribosomes, right? Those are there to construct proteins. So they read the message, basically a copy of DNA in the form of RNA, and then they use that RNA instructions to build proteins by connecting together amino acids to build that peptide bond. Yeah? They're filled, again, with that cytosol, that water-based jelly stuff, okay, so that thick viscous fluid, and that all is enclosed in a cell membrane and usually a cell wall as well. So bacteria will have an additional structure outside of their plasma membrane called a cell wall that provides extra protection. Right? Because if you think about it, these guys are just living anywhere they can, and they're not really in control of their environment per se. So that cell wall provides extra protection from the environment. And it's not uncommon also outside of that cell wall to have a glycocalyx layer. So that's just a layer of like sticky outer substance, a slime layer that basically either lets these organisms connect to each other or to stick to a surface. So that way they don't just get blown around by the atmosphere or water or something. So that's the big stuff inside of a prokaryote. Let's talk a little bit more about their cell membrane. Okay, most bacteria come in one of two flavors. They're either going to be gram-positive or gram-negative. And what that gram means, that means that if we treat bacteria with this gram stain, if they turn purple, that means they're gram-positive. And what's going on in these gram-positive bacteria is outside of their plasma membrane, they have a cell wall made out of a really thick peptidoglycan layer. So this is a combination of starches and proteins that creates a really thick layer outside of that plasma membrane. Okay, and these gram-positive bacteria, when we treat them with a gram stain, that gram stain basically can grab onto and attach to this really thick peptidoglycan layer and turns it purple. Okay, so if a bacteria stains purple when using a gram stain, it is a gram-positive bacteria. The other main type of structure that we see in the cell membrane of prokaryotes is the gram-negative bacteria. Okay, so here's the inner plasma membrane. We still have a peptidoglycan layer, but look how much thinner it is, right? It's much smaller. And now we add another layer on top of that. So we have the inner cell membrane, peptidoglycan layer, and then we have the outer cell membrane. So in gram-negative bacteria, when we treat them with gram stain, that gram stain can't get through this outer membrane. So it cannot bind to this peptidoglycan layer. So typically these bacteria, we treat them with something else and they end up turning pink. Okay, so if it's pink in a stain, it's a gram-negative bacteria, meaning it has this double layer right here. Okay, if it's purple, it's a gram-positive bacteria and has only a single layer of plasma membrane, but a really thick peptidoglycan layer. So some bacteria have the ability to move within their environment. They'll often have flagellum or multiple flagella, okay? And these are just long tails that they can kind of rotate in like a corkscrew type fashion. that allows them to propel themselves through the environment. So they have some proteins that are anchored inside their plasma membrane that extends out through the cell wall, and then they can basically turn those proteins using ATP to kind of fuel this motor, and that allows for a corkscrew type motion. They don't have any way to quote unquote see in their environment. They can't sense light per se, but they can detect some stimuli, specifically things like maybe where food is at, or maybe where other bacteria of their same kind are at, and they can generally move towards or away from a stimulus, but that's about it. So they can move, some of them, and they move by having these flagella that kind of propel themselves in a corkscrew fashion. This is going to be different than the flagella that we see in eukaryotes, which kind of go back and forth. Okay, so same idea, a tail, a cellular tail, but the way it moves is slightly different. All right, how do bacteria reproduce? So some bacteria can go through a generation time, meaning they can double their numbers in about 20 minutes. It's like the average generation time of E. coli is around like 22 minutes, I think. So yeah, you can get a lot of bacteria in a very short period of time, starting with just one. So bacteria reproduce through a process called binary fission. Basically, they copy all of their DNA, and if they have any extra DNA, sometimes they'll store extra DNA on what we call a plasmid. So plasmids are basically another circular piece of DNA where the bacteria keeps extra information, often helpful things like how to be antibiotic resistant, or how to break down specific sugars in the environment can be contained on that plasmid, and it can exchange plasmids with other bacteria. But it'll copy its main chromosome, and it will copy any plasmids that it has. Then those two separate. So this blue right here is the copied chromosome. This is the original chromosome. That two pieces of DNA separate, and then the cell divides in half. Okay, so they basically just expand, divide in half, and then we get two new bacteria. Okay, so we call this binary fission. We take one bacteria, copy all of its DNA, boop, divide it in half, and we get two new bacteria. So it's an exact copy of the original. Okay, so bacteria make exact copies of themselves to reproduce. But even though they're making exact copies, bacteria can do some weird things of mixing and matching some DNA with each other. One of the things that there's other ways besides this process of conjugation that we're talking about right here, but this is, I think, probably one of the more important ones. So bacteria can do this thing where, remember that plasmid we talked about? That plasmid that can contain helpful genes like how to be antibiotic resistant or how to break down or build certain enzymes, right? That plasmid can be exchanged between bacteria. So if we have one bacteria that finds a bacteria of an opposite cell type, okay, they form a mating bridge, or sometimes called a sex pilus, which basically is they just connect their cytoplasms together. It's just like a bridge, a tunnel that they can connect their cytoplasms together. And then the donor creates a copy of the plasmid that it has that might contain some helpful genes. And then it sends that copy of that plasmid through that bridge to the receiver cell, the recipient cell. Okay, so now this cell also has this helpful information that was in this plasmid. Okay, so even though bacteria don't go through sexual reproduction results in the mixing of genes, right? We have half the genes from our mom, half the genes from our dad. Even though bacteria don't go through sexual reproduction, they can still mix up their DNA with each other through this process of what we call conjugation by sending plasmids around to each other. Okay, so again, even though they seem really simple, and they don't go through sexual reproduction, they're just single-celled organisms. They are incredibly diverse. They're especially diverse in how they get their carbon and their energy. Okay, we're going to use some terms here that are probably going to be a little confusing to start with, but I want to break them down for us. So, like, for example, this term right here, photoautotrope. Troph basically just means to feed. in a sense. And these two prefixes, photo and auto, give us information as to the first prefix tells us how does it get its energy, and the second prefix tells us where is it getting its carbon. So in a photoautotroph, photo means light. Okay? So a photoautotroph gets its energy from sunlight, solar energy. And auto means self, but we can think of auto here meaning inorganic sources. So photoautotrophs use sunlight to build their sugars out of inorganic carbon dioxide. Okay, so these are things like plants, right? Or algae that photosynthesize. They use sunlight and carbon dioxide from the atmosphere to build the sugars in their body. Okay, so a lot of bacteria are photosynthetic. Cyanobacteria changed the face of the planet at one point in time when they were the dominant species, right? So they're using sunlight as their energy source with inorganic carbon source to make their sugars. Okay, so sunlight, inorganic, feeding is what photoautotroph means basically. Most bacteria, though, are going to be down here, chemoheterotrophs, meaning that they get their energy by breaking down organic compounds, chemo. They're breaking down chemicals, specifically organic compounds, by eating other organisms. And then hetero means that they also get their carbon from another source. So they also get their carbon from those organic molecules that they are consuming. Okay? So they get both their energy and their carbon to build up the molecules in their cells from other organisms, just like us. We are also chemoheterotrophs. So we eat food, and that's where we get our energy from. We also get our carbon source from that food that we eat. Okay, so we are also chemoheterotrophs. Some bacteria, though, might fall in some of these other categories, chemoautotrophs or photoheterotrophs. Most are going to be chemoheterotrophs, though, and some are also going to be photoautotrophs. But the whole point of this being is that they have a huge, wide variety of ways to get energy and to get carbon. So they can live in many different environments, and they can use many different sources of energy and many different sources of carbon to exist, which is part of the reason why they're so diverse, why there's millions of species of prokaryotes. And then lastly, what does they do in their environment? What are bacteria doing in their environment? What's their role, right? Some bacteria fix nitrogen. So nitrogen gas, remember nitrogen gas has 3 bonds between that molecule, between those 2 nitrogen atoms. That's rough. That's a hard bond to break. That's a really strong bond. And most living organisms cannot break up that triple bond between nitrogen. Okay? Some bacteria can, though. So they take that nitrogen gas in the atmosphere, they break apart that triple bond, and then they turn it into a usable form, usually nitrate or nitrite, which is super helpful for living organisms because now, once that triple bond is broken, that nitrogen can be used, especially for proteins or for nucleic acids. Those are very nitrogen-heavy compounds, right? So they can convert inorganic nitrogen into organic nitrogen, which is huge. Some of the only organisms that can do this, which is a really, really important ecological role. Most bacteria are also decomposers because they're those chemoheterotrophs, right? They're often eating dead organic material. So most of the decomposition that happens in an ecosystem is because bacteria are eating that dead stuff, like the leaves that fall on the ground or a dead organism that died out in the forest, right? Bacteria are going to be most of that. They're going to be the ones that are mainly responsible for that decomposition. And then again, recycling those nutrients, that nitrogen, that carbon, that oxygen, that phosphorus, all of those nutrients, all of those elements that are important for life. By breaking down that dead organism, they recycle those nutrients back into the environment to be reused by themselves and then by other organisms. Prokaryotes can photosynthesize, just like plants. Cyanobacteria are one of the major carbon capturing organisms on our planet that bring in carbon dioxide build sugars with it, and in the process release oxygen, which keeps us alive, right? And photosynthesis also feeds the rest of the planet, feeds the rest of the ecosystem that they're in. So they play a huge role in photosynthesis and carbon capture in our environment, in our ecosystems. Many bacteria are symbiotic with us and with other organisms, right? We have bacteria that live that we're coated in bacteria right now. We have Staphylococcus bacteria that are living on us and in us. We have E. coli bacteria living in our gut. We have tons of species, millions if not trillions of bacterial cells living on and in us. Most of the time they're not harmful though. They're helping us. They're either symbiotically living with us by helping us break down the food that's in our gut, or they outcompete potentially harmful bacteria that might want to cause us disease, right? The presence of population bacteria living on our skin right now might prevent a harmful bacteria from setting up shop and causing us disease. Okay, so bacteria are often symbiotic with other organisms living in or on them, where they get food from that organism and then they help that organism break down that food. A very, very, very small percentage of bacteria are actually pathogenic to humans, meaning disease-causing. So anywhere from like 5 to 3% of known bacterial species might be pathogenic. But part of the reason why we discovered bacteria in the first place is because they do cause disease, right? Things like bubonic plague or tuberculosis, those are all caused by bacteria. Okay, so they can be pathogenic, but we have drugs to treat them. Antibiotics are effective for now against most common bacterial infections.